very much for having invited me to speak in this seminar, and to thank the other organizers. I think this is a great seminar series initiative, and at least there are some advantages to the current coronavirus situation, so that, for me, it's really great to be able to speak in a seminar in San Diego while sitting at home in Switzerland. And so it's a great chance to see some people. So as Yusuf said, I'm going to talk primarily about some work that I've done applying topological data analysis in the framework of studying neuron morphologies, the shapes of neurons. I'm going to talk about how we can associate to a neuron a particular topological descriptor called a barcode, and how we can use these barcodes in order to classify these sort of tree shapes. And then talk about how one can go back, start with one of these topological descriptors, barcodes, and synthesize tree-like shapes that are associated, that have that barcode or very similar ones. And then I'll spend the last part of the talk discussing some of the mathematical statistical aspects of this. So I just want to say a couple words about the background image for the title slide. So this is actually a computer-done visualization of what are called pyramidal cells, excitatory cells from rat cortex. It's showing the great diversity of their morphologies, both in terms of their size, the branching structure, the curvature, and so on. Just give you some indication of the incredible diversity of neuron morphologies. And since the shape of a neuron is closely related to its function, it's really, really important to be able to characterize, to find good ways to describe.
So I said hi.
Good ways to describe this structure in order to better understand their function. Now, Yusuf asked me to say some words about the other work that I did, so I'll start with that, just to advertise this work that we did. This was work it was published in 2017, so it was really done in 2015 and 16, using topological data analysis in order to do high throughput screening of nanoporous materials for their performance properties. In particular, we were thinking about methane storage and methane capture. So this was in collaboration with colleagues at EPFL, Senja Bartel, who was a postdoc at EPFL at the time, and Pavel Vlotko in Swansea. And so I have a couple of articles, one published in Nature Communications in 2017, and the other the following year in the Journal of Chemical Theory and Computations, where we used very straightforward TDA methods to show that by looking at the shapes of the pores in these nanoporous materials, you could, and characterizing those using topological data analysis, you could sort of determine which materials might be worth synthesizing in order to optimize the performance parameters that you were interested in. So if you'd like, I can answer some more questions about this after the talk, but for the time being, I'm going to move on to neurons. So the outline for today is going to be first to talk about how you go from trees to barcodes. So when I talk about a tree, I'm going to be talking about embeddings of trees in 3 space. So if you're not interested in neurons, which is fine, there are lots of other interesting tree-like structures that one can be interested in studying. And I imagine that some of them are probably even relevant within material science to think about the different tree-like structures. And so we're going to see how we can use what we call the Topological Morphology Descriptor, TMD, which associates to a tree embedded in 3 space, one of these barcodes, they're called barcodes because they really look like the barcodes you see on products that are scanned at the grocery store, for example. And we use these barcodes, which are quite simple, to classify these neuron morphologies in ways that are relevant for understanding their function. In the second part of the talk, I'm going to explain how it can go back, start with a barcode, and synthesize in a reasonable and a biological way, in a way that's relevant for talking about neurons, different kinds of trees embedded in 3 space from barcodes. Solving inverse problems is in general, something people are interested in doing, and it's often a much harder problem than doing the opposite. And I think that I'm going to go into some detail about how this algorithm works and how we exploit the barcode in order to do so, because I think that by appropriate tweaking, one could probably arrange the synthesis algorithm so that it was relevant for a context other than talking about neurons. Then in the end, I'm going to talk about some different perspectives coming from sort of a more mathematical and statistical point of view on these constructions. So in particular, we're going to look at what are the possible geometric trees that can be associated to a barcode, and also some sort of stability results, like how much can these topological realizations, these tree realizations of a barcode change depending on how different the barcodes are from each other. This is a sort of question that is very relevant in these contexts of TDA when one is interested in how stable the particular invariants are. But before I launch into this, I need to give you a very brief introduction to neurobiology, just so we get some language on the table. I mean, I know I'm not talking primarily to biologists today. I know perhaps mostly neuroscientists or data scientists. So just a little hint of neurobiology. Here we have a cartoon of a neuron. So this doesn't look anything like what a neuron really looks like, but I want to talk about its main parts. So here we have the cell body, the soma of the neuron, which is where the information processing, or most of the information processing takes place. Here we have these finger-like structures here. These are called the dendrites. These are where the neuron receives its inputs. So electrical signals coming from other neurons. They enter through dendrites. There's some information processing that already happens with dendrites. Then the bulk processing seems to take place here in the soma. And as a result of this, you have the membrane potential of this cell here that can rise. And if it hits a certain threshold, the neuron does what's called spiking. It releases an electrical signal that will then travel down this axon here. So this is another tree-like structure that's sort of branching off the other way to the dendrites of another neuron. And the connection points from the axon of one neuron to the dendrites of another are called synapses. We're going to be focusing primarily today on dendrites because there are many more known examples that have been extracted in labs of what dendrites really look like. Axons are more mysterious because they're much longer, they're much more difficult to capture completely. So we have a larger database of dendrites to study. And so these dendrites are really sort of tree-like structures. And we're going to be interested in like how we can describe these tree-like structures here that are the dendrites. So onwards, how do we get from trees to barcodes? And I will remind you what barcodes are as well, or tell you what they are. So the question we're trying to answer is, how can we classify neuron morphologies in a reasonable way? So we have these structures, these tree-like structures, and we'd like to be able to say something about how to classify them, how to characterize them, how to describe them in a way that is relevant for their function. So here's an example of two of what are called digital reconstructions of neurons. So where these things come from is that in a lab, somebody will take a slice of brain tissue, a very thin slice of brain tissue, put it under a microscope, and then some poor PhD student or postdoc has to spend many, many, many hours peering into the microscope at this neuron and very carefully recording the three-dimensional coordinate, the 3D coordinates of quite a number of points on this whole tree structure. And then that is a way of encoding the structure of the neuron in a way that a computer can store and understand. And then you can retrace it in a drawing like this. So these are coming from actual neurons that were examined under a microscope and their 3D coordinates recorded. And so here we have the cell body, the soma, and what we're looking at is, this is what's called an apical dendrite. It's one of the dendrites that's extending towards the surface of the brain. And then we have a bunch of sort of very hairy roots here, and that's called the basal dendrite, which sort of just is this ball of roots around the cell body. So here we have these two neurons, and you'd like to say, well, they look kind of different, but how different are they? Can we say where the difference is and characterize it? That's the kind of question we're going to try to answer here. So what we did was develop an algorithm we called the Topological Morphology Descriptor to get there. So here's a cartoon of how this works. So you can think of this as being a tree embedded in 3 space. And what you do is you take your tree, which has this root R here, and you just take the tree and you break it up into its branches in a sort of principled way. You start with a leaf that's farthest from the root, and you just move progressively until you get to a bifurcation point. And at the bifurcation point, you break off the branch that is shorter. So where the leaf is closer to the root. So here we would break off this branch here and record it as a bar where the endpoints of the bar tell you how far, so one records the distance of the leaf from the root and then the bifurcation point. And you would continue on from there to the next bifurcation point. And here you say, okay, I need to break off the branch that is shorter. And that would be this branch here, which would then be recorded as a bar in this barcode. And you continue on this way until you get to this bifurcation point. And you say, okay, well, here I need to say where was this coming from? It's coming from here. I have to backtrack to yet another bifurcation. And so as I would say, okay, what would happen here? I would start at this leaf that's further out, this bifurcation point, I'm going to break off this branch here and record that as a bar. And so I would have a long branch that is going here. But since H is farther out than C, I would end up saying, okay, now it's time to break off this branch and record it as a bar here. And then the only thing that's left is a long, long, long branch going from the furthest leaf all the way to the root. So let's record that as this bar HR. This is how we get a barcode out of the tree. It's a very simplistic way, but it manages to capture both the combinatorial structure of the tree and enough information about the embedding to be interesting. Then one can take this barcode and translate it instead into what's called a persistence diagram, where each interval is in fact recorded now as a point in the plane, given by its coordinates. There's one point here that is above the diagonal. That's because here the leaf was actually closer to the root than the bifurcation point was. So there's this relatively simple algorithm you can apply to any tree embedded in 3 space. You could also generalize this to something more like a graph structure or a lattice structure once you fix sort of a choice of base point or reference point with respect to which you could do these computations, which could also be interesting in other less tree-like contexts. Now, when you do something like this, you would like the resulting barcode or persistence diagram to not be too sensitive to errors that might have been made in recording the actual distances or in recording the fine branching structure. So it turns out that if you think about the possible small errors you could have, for example, having made small errors of measurement here and here, or for example, if you had your measurements are more or less correct, but maybe you'd forgotten some of the very, very small branches. If the PhD student was particularly tired when he was looking under the microscope, he might have missed some things. But it turns out that you can quantify the fact that the topological morphology descriptor is in fact stable with respect to such small errors of reconstruction. If you're off by a little bit, it doesn't matter. The barcodes that you get will in fact be close to each other with respect to what's called the bottleneck distance. And I'm going to define this distance more precisely later. But just to say, it's not too sensitive to errors. So let's go back to our 2 original neurons that I had here, this one and this one. And what you can do is, if you're given one of these persistence diagrams, so it's a collection of points in the upper right-hand quadrant, you can associate to it what's called a persistence image. You get a persistence image from a persistence diagram by associating to each point in the persistence diagram a Gaussian kernel. And then you sum up these Gaussian kernels, and now you have a function on the upper right-hand plane, which you can then pixelize if you want. And now you have a matrix with respect to which you can do computations, which is represented here as this sort of heat map. So we have a lot of points here where it's red, fewer points where it's orange and green, and basically no points where it's blue. So the persistence, these are the persistence images associated to this neuron and to this neuron. And now we see very clearly where the differences are. The fact that we have a sort of a hot spot here corresponds to the fact that we have this tuft up here that we don't have on this side. We don't have any sort of hot spot here. And so by looking at the persistence images, we can see quite clearly the similarities and differences between these 2 neurons. And then if we sort of subtract this one from this one, we get this image here, and that's showing really emphasizing where the differences are between the 2 diagrams. So we applied these methods of associating 2 different neurons, first barcodes or persistence images, persistence diagrams, and then the associated persistence images coming by this Gaussian kernel process. That gives you objects to which you can apply statistical methods and machine learning techniques, for example, and then do different kinds of classification. So one of the classification problems we worked on had to do with classifying what are called pyramidal cells in rats. So these are exactly the neurons that I was showing on the very first slide. And usually when neuroscientists are classifying these neurons, they have a number of sort of standard methods to do it. One way is that you have an expert who has spent many, many years learning to recognize the different types of neurons, and he will do a subjective, just expert-based classification, looks at a neuron like this and says, oh, this is definitely a layer 5 thickly tufted pyramidal cell. They know these things from having seen them. Or people say, well, let's get a little more systematic. We associate to every neuron what are called different morphometrics that has to do with like how many branches it has, how many layers in the tree, how, different length measures and curvature measures and so on. And to each neuron, you can associate a vector of these different morphometric values, and then you can apply different sort of standard classification techniques to these vectors in order to cluster them and perform your classification. The problem is that the classification that you get strongly depends on which of these morphometrics you compute. And on the other hand, with the expert-based classifications, there were experiments done that showed that two different experts, even if they're both highly reputable experts, are not necessarily going to give you the same classification. And even the same expert on 2 different days doesn't necessarily give you the same classification. So it's something you have to be careful about. So it's good to have maybe a more objective classification method, which the Topological Morphology Descriptor provides us with. So we took a database of digital reconstructions of rat pyramidal cells, associated to them these barcodes and persistence images, and then did statistics, applied machine learning methods to the persistence images, and came up with a proposed objective classification, which we compared to the expert classification. And the expert classification, the subjective one, was in general very good. We made a few corrections, a better separation between the different types of what are called layer 3 pyramidal cells. So here we have the different neurons that are represented according to the layer of the cortex in which the soma appears. So this cortex has 6 different layers. And depending on in what layer you find the cell body of the neuron, you classify the neuron according to that. So these are the layer 3 pyramidal cells because their somas are in layer 3. And so we were able to, on one hand, both confirm most of the expert-based classification, but also to provide some clarification of this. For example, that really there aren't, in our opinion, 2 different types that are called A and B, but there's really sort of a continuum that contains both of these there, it turns out. So that was one classification task. Another interesting application, this was work that the lead researcher on this project, Lida Kanari, did with the team of Idan Segev at Hebrew University, where they were studying pyramidal cells from the human brain. And Idan Segev had the deep intuition that there should be a way to classify these neurons according to their electrical behavior, but using standard statistical and machine learning techniques was not able to come up with a convincing classification. Lida applied the TMD method to classify these human pyramidal cells by their morphology, from which the classification by their electrical behavior turned out to be more evident. And so there was a very nice sort of dichotomy into cells that were either thickly or slim tufted. And it turned out that there was a practically parallel dichotomy that became clear in terms of their electrical behavior as well. So another confirmation that structure and function are very closely linked in neurons. But it's interesting that, without sort of figuring out first what the morphological classification was, they weren't able to make this separation in terms of the electrical behavior. So how do we go back? So this is work that is currently under submission. You can find it on the bioRxiv if you're interested. So we want to go backwards, right? So we know how to take a neuron and break it up into a barcode. And now we'd like to be able to take a barcode and reconstruct something neuron-like from it. So why would you want to do that? Well, people, mathematicians, other people too, like these sort of inverse problems. But the real reason we were interested in doing so is because Lida is a collaborator on the Blue Brain project, and I work often with them as well. So the Blue Brain project is in the process of doing digital reconstructions of entire parts of, well, rat and mouse brain for the time being. Here we see one of their first major constructions, which is a microcircuit. It's part of the brain of a rat in the somatosensory cortex. So that's part of the brain that's responsible for processing incoming sensory input. You see the 6 layers of neurons here. So this is a very small piece of brain, only 31,000 neurons. But in order to populate such a digital reconstruction of the brain, you need a lot of models of neurons. And there aren't, and of a great variety thereof as well. And there aren't that many that come out of actual laboratory work. So in order to have some sort of principled reconstruction of synthetic neurons with which to populate such a digital reconstruction, it's a good idea to be able to have a machine that can like create populations for you that actually really look like the biological thing. So that was our motivation for doing so. So in order to understand the algorithm I'm going to describe, I need to tell you a little bit about how neurons actually grow. And if you have other examples in mind of tree-like structures that you're interested in, you can see what the parallels are with how neurons work and think about how you might modify the process I'm going to describe to fit whatever tree-like structures you're interested in. So there's the initiation of the neurite. So at what point do the dendrites or the axons, but we're focusing on dendrites, start growing from the cell body? Then there's the actual growth of the neurons. And there's the axon that has to find its path. And then branching. So as the neurites grow, at some point they branch, they bifurcate. And we're not going to talk about axons because I said axons are something people don't understand as well. We were going to focus only on dendrites. So when I talk about neurites, it's not going to mean only dendrites in this case. So here's a cartoon of how the synthesis algorithm works. So there's the stage of initiating the dendrites on the cell body. What we're really going to be interested in is the branching process. So how does this bifurcation take place? How do you determine when you actually bifurcate? How do the dendrites actually elongate it? What kind of path do we use? So these neurons are synthesized sort of stepwise process. And what I'm going to tell you about in more detail in the next slide is how we figure out what to do at each particular step in the process. Do we just continue? Do we bifurcate? Do we terminate? And between 2 steps in the process, what path do we go through? So here, I just want to point out that this is a description of what's going on in the bifurcation process in terms of a drawing for the time being. So if we're going to start with some sort of barcode, we're going to start with a barcode that has a longest bar corresponding to the longest branch. And so that one endpoint of that will correspond to the root. And then we start growing our tree. And then as we grow the tree, we sort of move along the bars until we say, as I get closer and closer to an actual endpoint of one of the bars, I have an increasing probability here that I will actually bifurcate. So I will bifurcate at some point, at the latest by this point here. So there's a stochastic element in this process. And then corresponding to this bar, I say, okay, as I approach this endpoint here, I'm going to have an increasing probability actually to terminate. So we have probabilities of termination, bifurcation and termination that are determined by the bars here. And there's a parameter lambda that determines this exponential probability. And this lambda has a strong effect on what the end result actually looks like. So I'll talk more in a moment about what lambda we need to choose. So a little more detail about the synthesis algorithm. First, we need to generate the cell body. There's a way of sampling this. We model it just by a sphere. We sample from some biological distributions. We sample how many neurites, like how many dendrites are actually going to grow out of this. And then for each neurite, we initiate it at a point that's determined again by some sort of distribution. And then we elongate each branch as a random walk, taking into account certain memory. It's a random walk of Gatson-Walton type. And then at each step in the process, we have probabilities to bifurcate and terminate that are determined by the bars in the barcode, as I briefly described before. And it's depending on the distance that you've traveled from the cell body, from the soma. So a little bit more about the elongation step, because people might be interested in what sort of random walks we're looking at. So because of the scale on which neurons exist, where we're looking at segments that be always of length just 1 micron. And then there'll be a certain direction vector saying, okay, in which direction are we going to head with our walk? And it's going to be based on a certain memory of where we've been before, a target that is chosen at the very beginning of the neurite, and just some sort of random value to get you there has to be some sort of randomness here to be able to capture the sort of random walk aspect of neurite behavior. And then these branches are each grown independently. So this is just to give some hint of what's going into the synthesis algorithm. And there are variants on this. One can certainly apply in other contexts, depending on what kind of trees you're interested in studying. So I said that there's this parameter lambda, which is telling us about how quickly the probability to bifurcate or terminate grows as we approach the end of one of these bars in the barcode. And so just to illustrate the effect of the different lambdas, so here we were looking at what kind of neurons, what kind of trees you synthesize from a given barcode. So you have one barcode, and from it you synthesize a number of different neurons. And you want to see how much variance is there in the family, how close is it to looking like what you would expect. And it turns out that if lambda is too small, you end up with these puny things that don't look anything like neurons. This looks a bit better. And then it turns out that you get sort of the best distribution, the closest to, that looks most like biology, when the lambda that you take is close to 1, where this is on the same scale as the step size. So lambda should be roughly on the same order as the step size. Right, biologically most reasonable choice of lambda. So just to show you what these synthesis neurons look, how close they look to the biological neurons when we've done this. So here, on the one hand, we have some excitatory cells from different layers, and on the other side, what are called inhibitory cells, the ones that are sort of calming down the action. And in this figure, the blue ones they're coming from the PhD student noting down all the points in the lab. So these are real biological neurons. And on the other hand, these are sort of the ones that we would get from synthesizing. You take this guy, you could take its TMD, its barcode, and from its barcode, you resynthesize a neuron. You see that's not bad. They match pretty well. And we can even make this more precise, like how closely the synthesized neurons actually match up with the reconstructed ones, so the ones that are actually biological, by saying, okay, well, I take one neuron that is here indicated in blue. I look at its associated barcode, its associated TMD, and then I run the synthesis process a number of times. So I create a whole population of new synthesized neurons from this one neuron by taking its barcode. And so what we see here in this figure with the original neuron itself, and then what you see is sort of a red shadow around that, is like the whole population of other neurons. So it makes like a whole shadow family around that. And here, in this persistence diagram, the blue points are the original persistence diagram, and the red points are the union of the persistence diagrams of the population that was generated from this one barcode. And we see that it doesn't wander too far from what we had at first. And here we have a number of these morphometric features that neuroscientists usually compute: the number of dendrites, the number of the lengths, the distances, and so on, terminations, bifurcations, all these numbers that they associate to them. And we looked at computed these values for this population of synthesized neurons generated from one barcode to see how closely it matched. And they match quite well indeed for most of them. So there's it's a faithful reconstruction. You can also reconstruct a population from a population. So here we had a whole population of layer 5 pyramidal cells that had been reconstructed in the lab. So these recorded by the PhD students. And then a whole population of neurons that were synthesized from the barcodes of these guys. And then here are a number of these different morphometric values. One is interested in computing, section radial distances, path distances, and so on. And then here we have this sort of violin plots, meaning that what we're looking at for the synthesized neurons in red and the biological neurons in blue, these are the distributions of values. So we'll see that their distributions match very nicely for the two populations. So it seems to do a good job. And just one point here is that often when people the neuroscientists have developed algorithms for synthesizing neurons, there are many different approaches to doing so, but they tend to sort of sample bifurcations and terminations from independent distributions. But with these barcodes, it enables you to couple via the topology the distributions of bifurcations and terminations. And when we do that, so here we have the biological reconstructions and we have matching synthesized cells. And we see, here we start with the original persistence diagram. This gives you you see with comparing the red points with the blue points that the persistence diagrams match well. If instead we take the bifurcations and terminations as independent probability distributions and sample them independently, it doesn't look nearly as good and the persistence diagrams don't match well at all. So really, the fact that the barcodes enable you to couple these distributions via the topology turns out to give you a better reconstruction, a better way of synthesizing trees that match your original structure. Okay, so a bit of more mathematical and statistical insight into what's really going on here with these two sort of inverse processes. So this is an article that we've just finished with Adélie Garin and Lida Kanari. And so I'm going to start by, I'm going to become a little more mathematical now and be more precise about my definitions. And this is where we'll start to see like how general this context is. It's really not just about neurons. So when I talk about a tree, for me, it's going to be a finite rooted tree. So I have a tree with a root, and it's going to be a tree that contains only bifurcations, because these are the sorts of trees that we come across in neuroscience. One could, I'm not sure that what I'm going to describe could actually be generalized beyond trees with bifurcations, but one might be able to think about it. In any case, we have a distinguished root vertex, and every other vertex of degree at most 3. And what I'm going to be thinking about are embeddings of such combinatorial trees in 3 space. And I'm going to pose one little extra condition that one can get rid of, but it makes life a little bit easier for what I want to talk about today. I'm just going to assume that the embedding satisfies one extra condition, which is that if I'm looking at, as I move along the unique path from a root to a leaf, that the distance to the root increases monotonically. So I'm not going to have the branch that's bending back. It's always going to be moving further and further away from the root in maybe a kind of twisted way, but we're moving away. This is a condition we can remove, but it makes this discussion a little easier. So I'm going to call T the set of all geometric trees. And I'm going to say that 2 geometric trees are combinatorial equivalent if they are embeddings of the same combinatorial tree, the same finite rooted tree. We're really going to be interested in the combinatorial structure, how it interacts with the geometry. Okay. So now to be a little more formal about what barcodes are. So a barcode is nothing but a finite multiset of intervals in the real line. So I say multiset because each interval may appear numerous times. In the case that we'll be interested in, each interval appears only once, so it's actually a set and not a multiset. But in general, a barcode is a multiset. We're going to be focusing on strict barcodes, which are ones where all the BI and DI are distinct. So that means that it's now a set and not a multiset in particular. I'm going to assume that the BI, which we call the birth times, we call this BI sort of the birth time of the interval, DI the death time of the interval. We'll assume that the BIs are ordered, the birth times are ordered, and that all of the DIs are less than D0 if I is greater than or equal to 1. So what that means is that I have one longest bar. So this would be my bar from B0 here to D0 here. And it contains all of the others. And here I see that the birth times B0, B1, B2, B3, B4 are all ordered like this. So it's just a way of imposing one extra condition on these barcodes. So here I have my barcodes with their associated numbers from 0 to 4. And one can associate a permutation to any barcode in the following way, following straightforward. Well, first of all, let me just introduce notation for these barcodes. So call B the set of strict barcodes, and Bn to be those strict barcodes that have exactly n+1 bars. So the +1 is because I always have this sort of extra bar, the biggest bar that contains all the others. So it plays a special role. And so there is a fairly obvious function from the set of barcodes with strict barcodes with n+1 bars to the symmetric group on n letters that takes a barcode to a permutation sigma b, where sigma b evaluated on I is just the number of j for which dj is less than or equal to di. So it's telling you what the order of the depth times is. So for example, here the associated permutation would be the one that sends 1, 2, 3, because there are 1, 2, 3 bars that are less than or equal to, with the depth time less than or equal to this one. Sigma b of 2 would be 1, 2, 3, 4. Sigma b of 3 would be 2, 1, 2. And sigma b of 4 would be 1. So it gives you a nice permutation this way. You can associate a permutation to any strict barcode. And we'll say that two strict barcodes are equivalent if and only if the associated permutations are the same. So what that means, you can think about what this notion of equivalence means. It means I can shrink the bars a bit. You know, there's some sort of affine transformations that I can apply here and not change the permutation type of the barcode. So the TMD now is just a map from the set of geometric trees to the set of barcodes. It's nice to think about it in a functional way like that. And we'll say that a particular geometric tree is a tree realization of a strict barcode if the TMD, the associated barcode of that tree, is exactly this barcode B. And given a barcode with bars BI, DI, what we'll call R of B is the set of combinatorial equivalence classes of realization. So the preimage under TMD of B is the set of all tree realizations, and then quotient by this combinatorial equivalence relation. So it's just the combinatorial equivalence classes of tree realizations. And it turns out that two barcodes are permutation equivalent, so they have the same associated permutation if and only if the associated sets of combinatorial equivalence classes of tree realizations are the same. This is actually not difficult to prove at all. And then we have a very explicit formula for the number of tree realizations of a particular barcode by saying that it's just a product for I between 1 and n of the numbers of j's, such j less than I, such that di is less than dj. So the number of switches that have transpositions that have been happening in the permutation barcode. This is what we call the tree realization number of a barcode. And this value here is the index of the ith bar. What it is, it's the number of other bars that are contained in the ith bar of the barcode. And if you take the product of those indices of the different bars, you get exactly the number of combinatorial equivalence classes of tree realizations of a particular barcode. So there's a reminder of what we just saw. And once we have this formula for this tree realization number, it's easy to provide an explicit number for the effect on this tree realization number of the following two actions, either adding a new bar to the barcode or transposing the order of two bars. These are things one could imagine doing with no problem. And so I just wanted to show you what happens to the tree realization number when you add a bar. So if you look at the 4 black bars here, the possible combinatorial types of trees that realize this barcode are given by these trees that are denoted in black here. If you look just at the black part of these trees, you see that they're actually combinatorially different. Now, if you add one more bar here, then it turns out that, for example, there are 3 different ways you could add the bar to this tree or to this tree or to this tree. And that's because this one, the index of this bar here is in fact 3. And so that's which corresponds to the fact that there are 3 different ways you could add a red branch to each of these black trees. So there's this multiplicative process that tells you what happens when you add a new bar to a barcode. So this is quite simple combinatorics, just some fun observations. So in this diagram here, I wanted to make the following illustration. So these are all the possible combinatorial types of tree realizations of strict barcodes with 4 bars, where we're always constrained by this elder rule, which was told us how we should break up a tree into its branches. So there are 1, 2, 3, 4, 5, 6 possible strict barcodes, and there are 1, 2, 3, 4, 5, 6 possible combinatorial tree types with 4 branches. And what we see here is there's an interesting relation between the possible permutation types of barcodes and the possible combinatorial types of trees. So for example, this very simple combinatorial type of tree can be a realization of any one of these barcodes. And this barcode admits any possible combinatorial tree type as its realization. But in general, the relation between permutation types of barcodes and combinatorial types of trees is relatively complex. One nice way to see this, if you like group theory a bit, is to look at the Cayley graph of the symmetric group. So here we're looking at the case where we have, this is the case n equals 3. So we have 3 bars besides the longest one. So the associated symmetric group is S3. And we have this nice relationship where we can, in the Cayley graph, the colors here are saying which transposition we're applying to get move between the different permutations. So what this figure is showing you here, the different permutations, the associated barcodes, and then the associated possible tree realizations of those barcodes. And you can see what happens to the possible tree realizations and as you permute bars here. So this is just showing the effect on the possible tree realizations of applying different permutations. And we see again, it's a very interesting combinatorial structure that is quite interesting to explore. So one thing you can say was, well, okay, what do we know about this tree realization number? So all we did here was just say, okay, let's look at, for example, here, something where we have 6 bars, and we look at all the possible permutations of 6 numbers. And here, so there are lots of them. So that's what they're just ordered here along the x-axis. And on the y-axis, it's just a histogram saying how many, on this axis, we're looking at the number of possible realizations, the tree realization number. In here, this is the histogram counting how many of these permutations we get with that particular tree realization number. So there's always only one, which has the maximal, which corresponds to this barcode that's ordered like this. And then the smaller the tree realization number is, the more possible permutations that have that realization. So this is just showing what happens as the number of branches or the number of bars in the barcode increases. And then you could say, well, okay, let's go back to thinking about the topological neuron synthesis for lambda equals 1, which is our biological value for this parameter lambda. And so we generated 1,000 trees with the topological neuron synthesis for each of the permutation equivalent classes of barcodes. So these are the permutation classes of the barcodes. These are the possible combinatorial types of trees. And then based on this neuron synthesis algorithm, we say, well, what is the distribution of the different combinatorial types look in the population? And what we see is that the topological neuron synthesis doesn't really have a very strong preference, maybe a slight preference for this type. If you look at here, any combinatorial type was realizable in principle, and each one is realized, and it's a more or less uniform distribution. So there isn't, at least when lambda equals 1, we don't have much, it's quite a uniform distribution of the trees you would get by applying topological neuron synthesis. However, if we choose other values of lambda, so here are 6 different combinatorial types of trees. If we choose values other than lambda equals 1, then the distributions look quite different. So this is showing for different values of lambda, what the distributions of the different kinds of trees look like when you synthesize a large population. So for example, when lambda is very, very big, that basically the only kind of combinatorial type you get is this one. When lambda is very, very small, then for some reason this one is overrepresented. So it's really, we see that the populations that you generate depend a lot on the value of lambda that you choose. And here is something that I find quite remarkable, that so we're going through this process, we're starting with these trees. It could be any kind of embedding of a tree in 3 space. You're going to calculate its topological morphology descriptor and, and then look at, well, how many possible realizations are there? So what we did here was the following. We said, well, given a certain number of branches or number of bars, what is the maximum number of realizations you could get? So this is something you can just compute combinatorially. It's like in factorial. Here we're taking the log of that, so we get a curve we can actually see. And that's what this red curve is. And then the points that are here in sort of yellow-green are taken by saying, okay, I'm going to choose barcodes at random with this many branches or this many bars, and then see how many possible realizations there are. And that was plotted here just by doing this for a very large number of barcodes chosen at random, what the curve of the topological realization, the tree realization number looked like. And then compared it to what you get by saying, well, I'm going to start with, on one hand, either these basal dendrites, which are these root-like things here, or with apical dendrites, which are these more tree-like things here. And I start with one of those, I look at its TMD, which is gives me a barcode, and I say, well, what's the tree realization number of that? And what you see is the basal dendrites don't look very different from something to be chosen at random, but the apical dendrites, they're quite distinct from something chosen at random. So there's something special about these biological barcodes. You can also pick this up by saying, okay, well, let's look at the set of all of these biological trees, so coming from dendrites with 30 branches. You compute the associated TMD, and then the associated permutations. And you say, well, what sort of permutations actually show up? And what this plot is showing you here was for each of those permutations, we plotted the point of an integer matched with its value under the permutation. And those were plotted here as red points. Now, if every possible permutation were equally represented among these biological barcodes, we would expect this whole thing to be red. But what we see is that there's really a concentration of the permutations that show up sort of close to the diagonal here. So there's something very special about the permutations that you can find as the permutations associated to the barcodes of actual neurons. We can also say something about the stability of this process. So you can start with a barcode, choose some tree that is synthesized in this way. So this is a stochastic element in this topological neuron synthesis. So it's not going to be just one tree, it's a whole family of trees, which is why I like to have like the power set of trees here. And then I take this tree and I compute its TMD. So it's like I apply TNS to this tree. So I start with a barcode, build a tree from it, and then take the barcode of that. And your question is, how far is that from the original one you started with? Well, it turns out that if they at least have the same permutation type, when you go up and then come back down, then what's called the bottleneck distance between the barcodes, and this is a definition of this bottleneck distance, the probability that this bottleneck distance is less than epsilon is this particular function of epsilon, which I can summarize briefly by saying that the probability that the distance between them is greater than epsilon decreases with epsilon. So there is some sort of stability in this process. You're not going to be too far away, at least as long as you don't actually change combinatorial type. There's also a sort of transposition stability. If you know that the ith death time is less than the jth, then the probability that after applying this process, these two will have switched places is actually decreasing exponentially with the distance between the two. So if the two death times are very far apart, it's very improbable that they will have switched after this process of synthesizing a neuron and coming back down with its barcode. So that this topological neuron synthesis provides a sort of approximate inverse to the TMD. And so here's some experimental results on this sort of transposition stability, where for lambda equals 1, we can see that, so this is the blue curve here, we can see that when the distance between the bars is large enough in the order of microns, there's very little probability that the two are going to change place. It's only when the distance between them gets very small that we start having these sort of switches going on. On the other hand, it's not such a good left inverse, because if you start with a biological tree, so coming from a neuron, you compute its associated barcode, and then you look at 100 different instantiations of what you get by synthesizing new neurons from that barcode. And then you, again, come back down to compute the associated barcodes to those 100 trees, and you look at the associated permutations, and you see that there is some variation going on. You can still switch permutation types. You don't see it so much in the persistence diagrams themselves, but if you start looking at the permutation types of these barcodes, then you see that there is some variation. So you can actually change things around. So the questions we're interested in considering now are some on the order of biological. And this is where, if you're interested in trees for another reason, you could be interested in analogous questions. So clearly, not all barcodes are going to show up in the TMD of actual neurons. So how can we characterize those that do? Which particular permutation equivalence classes are actually realizable by geometric trees? And then we'd like to be able to exploit those particular descriptors in order to say, well, can we distinguish between morphological types of neurons, or from neurons coming from different brain regions, or from neurons coming from different species? So these are really, I think there's a lot of potential for interesting biological insight in terms of finding some characterization or description of the subspace of which barcodes are actually biologically realizable. And then there's a sort of cellular, or you can decompose these spaces of strict barcodes in terms of the permutation equivalence classes. And there's an interesting study going on by Justin Curry, Jordan Deschamps, Adélie Garin, and Brendan Mallory, who are trying to understand in detail the decomposition of this space, which has actually something to do with permutahedra and cones on permutahedra. There's interesting stratified spaces going on here. And I think there's a huge number of interesting things to explore there. So there's both the biology and the math, which are highly non-trivial and interesting in this context. So it's been a great pleasure to work with a number of remarkable mathematicians, neuroscientists, computer scientists, data scientists on this project. I'd like particularly to mention Lida Kanari, who's been the lead researcher on all of these projects and who's done fantastic work. And it's been my pleasure to tell you this story. I hope you enjoyed it, and I'd be very happy to answer any questions.
Thank you, Kathryn, for this excellent talk, and it's really interesting. So now you can, the audience, feel free to ask questions. You can just type the question in the chat box, or you can just unmute yourself and ask. So, okay, let's see. Questions? So, okay, let me actually start. I mean, I have many questions, but let me start with one. I really like the, yeah, no, go ahead, Bay.
It's okay.
Go ahead.
Yeah, sorry. Sorry, I just have a quick question. In the synthesized process, it feels like the geometry also looks really well. How do you decide the endpoint when you're growing this neuron? Because, you know, if I use just a random walk, I have this embedded R3 space. I can kind of grow it any direction I want. So it feels like in the synthesized process, you also have a guidance. And I think I missed that part. How do you guide the endpoint it's going to terminate in the synthesized process so that geometrically they all look very similar?
Right, so that comes from, let me just go back to something about the description of the synthesis. Here, that should be good. Right, so in the process of growing, so once you've decided whether or not you're going to bifurcate, suppose you're not going to bifurcate, you're not going to terminate, and so you just continue. So there are different components determining this. So this is what's called a Guts and Walton tree. So when you're growing it, the direction, so you're building it segment by segment, and you want to figure out like, where is this segment going to go? And so that's determined by a vector that's given by some sort of memory vector that remembers the previous direction vectors. But using it's a weighted sum where the weights decrease as your distance increases from where you are currently. You've chosen some target direction from the beginning of your branch. And so this is something that this is data that's coming through from biology, for example. And then you've chosen some sort of, at each stage you're choosing some sort of random vector where you're sampling uniformly from 3 space at each step. And so those are the 3 components, and then you weight those accordingly. Now, one figure that I don't have, but that you can find in our paper on bioRxiv, where Lida tested like various combinations of rho, tau, and mu, and you can see what happens when for example, if rho is too large, then you really get something that's just a random walk. It doesn't look anything like a neuron. If rho is 0, then you get something that is way too straight, and also there's not enough variance in the population, because we really want to have a population that has some variance. So it looks more like you want to, when you're doing this sort of reconstruction algorithms for a whole network, you really want a population that that is non-uniform. You have like a lot of different components, and so you need that random component as well. So it's quite interesting to see what the result is when you vary this rho, tau, and mu.
Thank you.
You're welcome.
Are those rho, tau, and mu learnable? I mean, if I have a sort of training set of barcodes, can I learn these 3 parameters by just reconstructing the trees and computing barcodes, find the difference? And
I suppose you could actually. So that wasn't the way we did it. It was more like just a parameter scan to say, well, where do we get the best fit? But I don't think Lida actually did it as a learning process. But I imagine that one could, and that's probably a good way to proceed.
Because from your initial slides, I immediately went to autoencoders. Like you have TMD, which is the encoding part, and then TNS, which is sort of the decoding part. And can I learn the entire thing?
That's a really interesting question. I don't think we've tried doing something like that. That's a really good idea.
Yeah, go ahead.
I had a question about the barcodes. So does the barcode not encode where the shorter segment bifurcates from?
So it does. Here we go. Right. So you mean when you're going along here and determining when it bifurcates, or? No,
from which branch? For example, the shortest segment, it could have branched from
I hear you. 3. This bar here, where it's going to branch from.
Yeah, sorry, the shortest bar.
All right. So, I mean, these 2 are sort of in competition. So it could branch from here or it could branch from here.
Okay.
It's not.
Yeah.
It just
biologically, it's not.
There's no. It could branch from either one. Yeah, and that's why when we're doing, and I talked about this uniform probability.
Yeah.
Yeah, so that's because when we were, I think that is worth revisiting. Here we go. Right, so here, this 1, 2, 3, right? So this is the barcode that is like this. It's the one that's sort of perfectly ordered like this. And so that gives you the most flexibility about where you bifurcate from. So the fact that each of these combinatorial types is basically uniformly represented means that this little purple branch here, it could sort of bifurcate off of anybody it wants to. And that's, and yeah. So it's not determined, no.
Okay.
So it's 11, so those who need to leave, feel free. But if you have questions, maybe Kathryn can stay a bit long, a couple more minutes to help to address.
Be happy to.
Oh, maybe a naive question.
Hi, Carlos.
Yeah. Well, yeah, if you take a geometric tree, I can imagine another natural way to create a barcode out of it. We just take a height function and just keep taking sub-level sets and counting how many connected components they have, as if it were a function, essentially. Would this give less information than the TMD method?
So we did, I believe, I mean, it's a similar kind of thing. So when you're doing a height function, what we're doing is we're doing a more of a radial height function, if you will. Instead of doing a height function like this, doing a radial function like this. So there was in the paper that we wrote about the classification of the rat pyramidal cells, we tried a number of different ways actually of analyzing the tree of what we used as a distance function from the root. And and looking at it from various perspectives. I don't know if we actually looked at just looking at the height for a particular value, although maybe one of the methods we considered was equivalent to that. But it turns out that really using either the radial distance from the root or in some cases the path distance within the tree is best. I mean, you try and so I don't have a proof of that. That's more like that's what a lot of experience with many data sets has shown us.
Thank you.
You're welcome.
So other questions? I think Faye also have another question here. Faye, you want to just ask?
Yeah, sure. So just exactly at the slide that Kathryn kind of posted, in this kind of picture in the empirical distribution of a combinatorial types, there seems to be those missing areas, right? Like you made a statement saying that the distribution seems to be much more complicated. Is there any intuitive explanation of those missing sort of tree realizations in this table that you're showing on this slide?
Like here, maybe this is even better.
Yeah, exactly. Yeah.
Yeah. So, I mean, this is basically the problem is the elder rule, right? The fact that you have to, at any bifurcation, you break off the younger or shorter branch. And that's basically what's forcing the non-existence of certain combinatorial types there. I think that's one of the main things going on. Yeah.
And then from the explanations that this doesn't really change the overall distribution because the order rule doesn't, in some sense, kind of force too many empties. Because I would naively think that it would be a distribution where everything is pretty uniform throughout this whole table of distributions, but the order rule has forced some non-existence of realizations.
That's right.
Is this pattern kind of generalizable that, you know, if I increase the number of branches and so on, is there like a generalizable rule kind of predict this distribution? I guess that's where I'm getting at.
Yep. I mean, that's basically what's going on here, right? So we're looking for the different possible tree realization numbers, like how many permutation types have that particular realization number. And so you can, and then this also, let's see, what I mentioned here, right? That you can exactly figure out what the effect on the tree realization numbers of adding another bar. And that's particularly what's going on. Like when you add a bar, you just have to say, okay, what's the index of that bar? How many other bars does it contain? And then boom, you know how to calculate, well, at least how many combinatorial types of realizations there are. And then as I showed in the next slide, you know, you can see, okay, here I had the 4 black bars, which correspond to the 3 possible tree realizations that are in black there. And then for each of those, when I add the red bar, there are 3 possible ways I can attach that red bar. So you can actually build this up.
Okay, so there's basically a sort of incremental kind of prediction just following the current formula. Mm-hmm. Yeah. Okay, cool. Thank you.
You're welcome.
Yeah, this is great. If I can, just 2 quick questions. I mean, those who has to leave, feel free to do so. I mean, I really like, I think this synthesis work is really interesting. You basically now have a generative model for the neuron trees, right? Right. So a quick question is that what can you do with this generative model? And then more on the theoretical side, you have this really nice result on stability respect to like when you come back again, kind of between the generated tree and the original one. Does that imply that in the end, the distribution of your generated tree kind of matches the original distribution? I mean, does it lead to that result? Because you have this condition where the combinatorial structure has to be the same.
Right. So, so the first question was about what do we do with the generative model? So, fortunately, I have an extra slide. Haha.
Yeah.
There we go. So one thing you can do is say, okay, what if my like, what if branches start shrinking or I start losing branches? And then you can say, okay, well, how does that affect the overall structure? And we're particularly interested in taking neurons and in building networks out of them. And you could say, well, if my neurons start being deformed in some way because they lose branches or they shrink. I mean, shrinking of neurons is something that happens when people are stressed, for example. It's known that stress causes dendrites to shrink. You say, well, what is the effect on how your brain is connecting up under those conditions? So one experiment that we did was to say, okay, well, what happens if I either start losing branches or they start shrinking? And then you can see what in this here, we see, okay, we're supposed to start losing the blue branches and then so on, sort of like, which points are we losing? And this is more like what's happening as I shrink things. And you can sort of see what's happening to the persistence diagrams. But then what we're doing here is saying, okay, let's build a network of these guys and see if we look at that as a graph, what sort of simplices do we find in different dimensions? We look at the associated flag complex, in this case, directed flag complex, and look at what happens to the distribution of simplices. And this is saying, okay, well, as I cut more and more branches, I have this number of simplices that completely dies out. Or if I shrink them, in fact, shrinking has a less dramatic effect. It has an effect, but it's less dramatic and so on. And so it's really interesting to see what happens. So that's the kind of game we can play. We can say, okay, we have the synthesis, and now we can play with the barcodes and see what the effect is on the trees.
Really interesting.
Now, in terms of what happened about this stability result, yeah. So what it means is that there is that you probably have there's probably some topology or some metric you can define on the space of trees, so that if you apply the TN, if you start with a tree, you apply the TMD, you come back with a TNS, then you're going to be within some sort of reasonable ball of where you started, and that we can probably, maybe someday we can even prove a theorem like that, but that's related to the results I was showing that had to do with the distributions of these different morphometric values, where it matched very nicely with the actual biology, like this one, for example, where these sort of morphometric values of the populations were very similar distributions.
Thank you. I mean, this is really interesting. Thanks. Is there anyone else? Any other questions? If not, if you can unmute yourself maybe and join me to thank Kathryn. Most of them are already gone at this point. Really, thank you for the fascinating talks.
Thank you.
Okay, thank you, Kathryn. So
Thank you.
yeah, I hope to hopefully the COVID situation passes and we get to see each other in person.
Thank you.
Okay.
I would appreciate it. I hope it would be a pleasure to visit you sometime.
That would be cool.
It's great to see everybody.
The weather is nice.
For...
All right then. Okay, thank you all for coming. Yeah. Bye.
Thank you. Goodbye.